It's always a great deal of excitement when you go to a totally different environment, particularly an area that we've not been able to get to before. This was the first time to get down to a depth of 4,000 meters, particularly over a lengthy area along the Australian coastline. So the fishes that come up from those depths are quite different from the ones from shallow water. So it's very much a discovery for people like myself who go on these trips.
The aim of the workshop is to identify the species that were collected as best as possible using a range of tools, including genetic barcoding, X-ray vertebral counts, and then also using microscopes to look at particular features of the species. And we'll also use the Australian National Fish Collection here as a resource to compare other specimens collected from shallower and other areas around Australia. We have over 3,000 species from Australian waters in the collection, and we have additional species from nearby areas in Asia that don't occur in Australia. To confirm the identity of the species of the fish, we need to ideally compare the specimens to other specimens that have a known identification. So that's why we compare all specimens to other specimens in collections. And also especially we need to compare the information we've collected from these animals to what's published in the literature, so that we can use as many different techniques to be as reliable as possible in our answer. People's ideas of what a species is changes, and what an identification changes. So what one person called a species in 50 years ago can be totally different to what people are calling it today.
The collections here at CSIRO, and like those that we have at Museum Victoria, are made up of specimens acquired through our field work, and they're retained and preserved for scientists around the world. Without that material, we're unable to compare what we have in Victoria around Australia with what occurs in other parts of the world.